Good morning, everyone, and welcome to the 2007 series of caucuses. You know, in the past 17 years, we had many caucuses on Alzheimer's, obesity, breast cancer, and we always look forward to having your office make a suggestion to us as to what topic you might want to hear next. You know, we had our four chairmen of our caucuses, Rush Holt, Lois Capps, Brian Bilbray, and Mike Castle, really worked on doubling the NIH over the last several years. And this year, like so many years, we're really caught up in difficult fiscal constraints. As you all know, there are letters floating around, dear colleagues, initiated by Congressman Markey and Chris Shays on the House side, Senator Kennedy and Orrin Hatch on the Senate side, and they asked for increasing the NIH by 6.7% because the NIH has been flattened. And we hope that you will be paying attention to those letters in your offices and doing all you can to support the NIH, because I think working together, we can sustain the research program. Today, we're going to learn about how synthetic biology builds on advances in molecular cell and systems biology and seeks to transform biology in the same way that synthesis transformed chemistry. Synthetic biology is used to create inexpensive, effective anti-malarial drugs. Currently, malaria infects 300 to 500 million people and causes 1 to 2 million deaths each year, primarily children in Africa and Asia. One of the principal obstacles to addressing this global health threat is lack of effective, affordable drugs. The development of this new technology will eventually reduce the cost of combination therapies significantly below the current price. We're privileged today to have a renowned scientist in the world that has worked on this issue and has made many discoveries in regard to malarial drugs. Dr. J. Keasling is from the University of California at Berkeley. He's the director of the Berkeley Center for Synthetic Biology. Dr. Keasling obtained his PhD in chemical engineering in '91 from the University of Michigan. His MS in chemical engineering in 1988 from the University of Michigan, and his BS in chemistry and biology in 1986 from the University of Nebraska-Lincoln. He is a well-known scientist. He has written countless treatises and done much research in this area. It's a great pleasure to welcome Dr. Keasling.
Thanks very much. It's a pleasure to be here. Today I'm going to talk to you about engineering microbes for production of anti-malarial drugs and the use of synthetic biology to do that. But before I do that, I want to just tell you a bit about the disease, because it's an incredible disease in terms of the number of lives lost and the extent to which it affects those that have it and don't die from it. It's caused by Plasmodium. Plasmodium is a single-cell organism that infects the red blood cell and consumes hemoglobin so that the cells can't carry oxygen. It's transmitted by the Anopheles mosquito. In green, I show here the countries in the world that are most afflicted by malaria. It's primarily around the equator in those countries that are very poor. At any one time, between 300 and 500 million people have the disease. 1/4 of the world's population is at risk of getting the disease, and every year between 1 and 3 million people die of the disease. 90% of them are children under the age of 5. And of those countries that are most afflicted, shown here in green, it reduces their gross domestic product by roughly half. There are drugs that have been widely used and abused, so much so that, in fact, they're no longer effective in treating malaria. They're based on quinine, and this is a derivative called chloroquine, which was actually one of the better drugs. It was used widely, so much so that Plasmodium became resistant to it. And these dots on this map show regions in the world where the drug is no longer effective. Throughout the world, this drug is no longer effective. It's cheap, it's available, but it doesn't work. And it's still being distributed. This is a plot of the deaths due to malaria over the last 100 years. And you can see that it was quite high and then dropped precipitously because of the use of chloroquine and also the use of DDT, which is a pesticide that kills mosquitoes. Now, the U.S. decided to ban the use of DDT and encouraged other countries to do so, and there's really no other pesticide that's available. And because the drugs no longer work, Plasmodium is resistant to them, and because there's no DDT, deaths are rising due to malaria. There is a drug out there that's a miracle cure. It's called artemisinin, and it comes from this plant, Artemisia annua. It's an ancient Chinese herb, and it goes back many, many years, which I'll tell you about in a minute. Now, the drug is produced in little oil sacs on the leaf of the plant. And originally, when this was discovered, and in fact, even today, it's extracted from the plant, now using solvents. Prior to that, when it was first discovered, by boiling the leaves and actually extracting it into a tea. It goes back as far as 168 BC, where there are writings of its use in some tombs of the ancient Chinese rulers, first to treat hemorrhoids and then later to treat fevers. And was largely forgotten until the Vietnam War, when drugs were needed for Chinese soldiers in Vietnam. And it was rediscovered from the literature, and the active ingredient was purified. And it's this drug called artemisinin. It's in a family of natural products. These are molecules, chemicals that are naturally made by plants, and we use them in all aspects of life today. Lycopene is a carotenoid. We find these in a lot of vitamins. Many of your vitamins contain lycopene. It's an antioxidant. Menthol is, of course, a fragrance. Many of the perfumes you wear are due to this family of natural products. Taxol, the anti-cancer drug that comes from the Pacific Yew, is in the same family, and even some bad things like cholesterol. Now, the problem with artemisinin is its cost. It's an extremely effective drug. If you buy it as a traveler, you might pay as much as $20 to $40 for a treatment. When the World Health Organization actually recommended it as the drug of choice for treating malaria, they negotiated a price of about $2.50 with the drug makers that are currently making it. But the problem with that is it's still too expensive by about an order of magnitude. People in these countries where malaria is endemic are living on less than $1 per day, and their governments are spending less than $4 per person per year on health care. So they can't afford the drug. What's more, malaria is unlike many other diseases. You can get malaria, be cured of it, and then get it again and again and again. And this is one of the real challenges in developing a vaccine for malaria, is that the Plasmodium doesn't present anything to your cells, so it's very difficult to develop vaccines. Therefore, you need much more of this drug than you normally would, because you have to use it again and again and again when you get the disease again. So the World Health Organization has estimated that we'll need 700 tons of this annually. And we have nowhere near this amount. Right now, it's being produced in small mom and pop farms in Southeast Asia. They harvest the plant and extract it with the only solvent they have available to them, gasoline. In the old day, a few years ago, before the introduction of unleaded gasoline, there was a real contamination problem. And then they sell it to pharmaceutical manufacturers that incorporate it into the drugs. When the World Health Organization actually recommended this drug as the chosen drug for treating malaria, they started withholding the crop so that they would get a better price for it, because there was a demand. And so the price began to rise. So my lab has been working on engineering microbes like E. coli, the organism found in your gut, to produce this drug, and to produce it in very large scale from a renewable resource. We take basically something very simple like cornstarch or corn syrup, use the metabolic reactions inside the cell, and we actually incorporate some alternative chemistry inside the cell, and the microbe actually spits out the drug, or a precursor to the drug, that we can then formulate into tablets. This is much like building a chemical factory inside a microbe. In a chemical factory, you of course would have reactors and distillation columns and pipes connecting them. We have the same things really in a microbe that does this chemistry. The chemistry is actually done by enzymes that catalyze these reactions inside the cell, and those enzymes are very specific. So you don't have to purify each reaction step in order to go on to the next step. So it's a very efficient chemical production plant, and I'm going to talk in some more detail about how we actually build that microbe to produce these chemicals. The first thing you do if you want to produce a specific chemical like artemisinin that's produced in this plant is you find the chemistry inside that plant that catalyzes the reactions to produce the product you want. And then you take those reactions and you put them into the microbe. And the way we put them in the microbe is we clone the genes that encode those enzymes, those reactors that I just talked about, and express those in a microbe so that the microbe uses chemicals that it normally would use to make the cell and reroutes them to produce this anti-malarial drug. Now, we don't have to get the genes exclusively from this plant, Artemisia annua. We can also get them from like organisms or organisms that are very different, like yeast, and I'll talk about that in a minute. So, once we've identified those pathways, as I said, we clone the genes out of the microbe, and hopefully those genes will then be expressed to produce the enzymes that produce our product. Now, when we started this project, we first started out with the first gene in the pathway, and we put it into the microbe, and the production levels were absolutely miserable. This plot here at the top is our target, and if you count here, this is 6 logs, 6 orders of magnitude between where we are and where we want to be, and that seemed almost insurmountable when we started this research. So one of the primary problems is that when you take genes from an organism like a plant and put them into a microbe, the microbe rejects the genes. Just like when you put a foreign organ in your body rejects it. Well, the same thing really happens when you put a foreign gene into a microbe. It can recognize that it's foreign and it rejects it. So the way around that is you recode the gene. It would be much like taking an organ, a foreign organ, and recoding it so that your body couldn't recognize it. And in this case, we make it look like a microbial gene. And then we synthesize the gene, and now you can actually send out and have whole genes synthesized. They use a reaction called the polymerase chain reaction, but it allows you to extend the gene. And when you do that, you get a huge bump in productivity, in this case 140-fold, but we're still 4 orders of magnitude away. Now, one thing you might want to do is increase the production of precursors to that product you want, but actually using the cell's native pathways, you don't get very far with that, only about another 3-fold. So we decided to take another lesson that we had learned, and that is this molecule is similar to cholesterol. And as it turns out, yeast, like you and me, has a cholesterol problem. It produces more cholesterol than it needs. It's a molecule called ergosterol, but it has a very efficient pathway. Just like you and I have a very efficient pathway for production of cholesterol, it has a very efficient pathway for production of its cholesterol called ergosterol. So what if we take that cholesterol pathway and give E. coli a cholesterol problem? And we did that. We took that pathway, and it actually allows us to reroute the chemistry very efficiently to this final product, and we got another 100-fold bump in production. Now I want to talk about the final step here. We do all of this in large tanks, and we blow oxygen through there because the microbes need oxygen in order to survive. And this molecule that we're making is a hydrocarbon. It's like gasoline a little bit. And we all know that in the absence of a spark, if you blew air through gasoline or gasoline and water, it would all evaporate out very quickly. But if you put an oil on top, you can actually collect all of that chemical inside the reactor. And you find out that you've got about another 100-fold. So we've just about hit our target. Now, up to this point, I've only talked about producing this molecule. We need some more chemistry to produce the product we really want. So we've got to go into the plant again and find the rest of that pathway. Well, when you're going into a plant where there's no genome sequence, many organisms have a genome sequence, and this allows us to very efficiently pull genes out of those organisms. But this plant doesn't have a genome sequence, and its genome is very large, and it would be very expensive to do this. So rather than sequence its entire genome, we used the genome of organisms that is known, sunflower and lettuce, for instance, and they make like molecules. And we thought, well, if they make like molecules, they might have similar genes, and we can use that similarity to pull the genes out of this organism. And so we, in fact, pull those genes out, and it actually allowed us to produce our final product. Now, as it turns out, this final product like it's toxic to Plasmodium, the organism that causes malaria, it's also toxic to the organism E. coli and yeast that we've engineered to produce it. But they're smarter than Plasmodium. They actually have pumps in the membrane, and they can pump this drug outside the cell so that it doesn't accumulate inside the cell and it's not toxic to them. So they actually pump the drug outside the cell and purify it for us. So this is the purification part of our chemical plant. Now, here's what I've just shown you. We've produced this precursor artemisinic acid. Now, that's not exactly what's formulated into a drug. The next steps you do are basic chemistry, chemical reactions outside the cell to convert that into all the different derivatives that are currently put into drugs. So artemether and arteether are drugs, molecules that are put into drugs for treating cerebral malaria. They cross the blood-brain barrier very rapidly, so they can actually attack the Plasmodium in the brain. And I was speaking to a doctor at Michigan State University. She spends half of her time at Michigan State and half of her time in Malawi. And she actually does autopsies on children who've died of malaria. And she said that she's seen many cases where they come into the hospital with malaria and they're essentially comatose. And they take artemisinin, and the next morning they wake up and they're playing as if nothing had ever happened to them. It's really a miracle drug. But the problem with these drugs is that they can be toxic to children because they cross the blood-brain barrier very rapidly. This is a drug we're targeting, artesunate. It's actually been tested and used by the US Army, even though this doesn't have FDA approval. So it's actually quite effective in treating malaria. Now, we put together a team to actually get this project done because we don't want to just stop at the scientific research. We actually want to get this out to people who need it. The work I've just described happened in my laboratory at Berkeley. A small company called Amyris Biotechnologies grew out of my laboratory, and they're actually going to be doing the scale-up, taking the bug from shake flask to large-scale reactors. We predict that we'll need about 50,000 liter tank producing this drug 24/7, 50 weeks a year, to produce enough drug for the entire world. Now, considering the size of the problem, that's actually a very small solution. And we've paired up with the Institute for One World Health, the world's first nonprofit pharmaceutical in San Francisco. So the technology goes from my lab to Amyris, where they optimize it, scale it up, and then to One World Health, where they find a large-scale contract manufacturer to actually produce the drug and distribute it to people in the developing world. And we hope to launch this product at the end of 2009, early 2010. And when we do so, we hope to lower the cost of the drug by an order of magnitude. And that's what the economic analysis tells us. Now, what's so nice about this technology is it's a platform. And that means that you can take a few of the genes out that we've used to produce this anti-malarial drug and put in other genes to produce other drugs. For instance, this is Taxol. It comes from the bark of the Pacific yew. It took 30 years from its initial discovery to get this out to people. Part of it was the source of Taxol. Taxol is produced in this bark of this tree, and it takes one 100-year-old tree to get enough Taxol to treat one cancer patient once. You need more treatments than there are Pacific yews in the world each year. So you can't get it from Pacific yews. They're protected species. As it turns out, a precursor to this drug can be found in the pine needles, so they can rake up the pine needles and derivatize it. But with this technology, we could take the genes, and almost all of the genes for this incredibly complicated molecule have been discovered in this tree. We could take just one sample of that tree, get the genes out, put them into a microbe, and get the microbe to produce this drug. Now, it's not at all trivial, and that would take years of optimization to work. But that's the power in having a platform, that you can adapt it for other drugs. Or say this drug. This is eleutherobin. It's found in this coral. As it turns out, it was going through clinical trials, but they needed more of the drug to take it through stage 3 clinical trials. The country around which this coral was found wouldn't give the pharmaceutical manufacturer access to the coral, and so it was dropped from clinical trials. It might have been a great drug. With this technology that I've just described, in theory, you'd only need 1 or 2 or a few corals, just enough to get the genes out, put them in the microbe, and get the microbe to produce this molecule. And this is a molecule now we're working on in the laboratory. It's called prostratin. It was used by healers in Samoa to treat hepatitis. They actually stripped the bark off of this tree called the mamala tree, boiled it, made a tea, and it was effective at treating hepatitis. Well, an ethnobotanist named Paul Cox actually went there, learned how to make the drug, took it back to the NIH where they purified it, found the active ingredient, and it's called prostratin. It's a relative of artemisinin. What's so interesting about this drug is it actually drives HIV out of senescence. So it doesn't kill HIV, but it drives it out of senescence. And then you can hit HIV with these combination therapies that we've already developed. This is actually just launching into clinical trials. It just made it through tox tests. We hope to have the genes cloned for this pathway to make this drug if it survives clinical trials, because we don't want them to cut down the entire rainforest in Samoa to get the trees and then plant it over with more trees. But this would be an incredible source of income for the Samoans if they did. So we actually went to Samoa and wrote up an agreement with the tribes people, the townspeople in Samoa, where we would get access to the trees in order to clone the genes out. And in return, any money that UC Berkeley made from this through patents, we would give half of it to these people in Samoa, because it's really their intellectual property. And this is an agreement we signed with the Prime Minister of Samoa. But we don't have to stop there. We can actually now use this platform to produce biofuels. And this is a project we're launching into now, where we actually use many of the same chemical reactions, but the final decorating steps we change. And we can produce things like alternative biofuels, things that could be used as jet fuels or as diesel fuels. And this would eventually go into a process that we're now using for producing ethanol. Now we take corn, turn it into starch, use enzymes to turn that into sugar, and microbes like yeast to turn it into ethanol. Well, we could actually now substitute these microbes and use that same sugar to produce alternative biofuels. So it actually expands the biofuels industry. Or even farther down the road, we can actually substitute in something like cellulose and use that as the starting material to produce these biofuels. And to actually launch these projects, Berkeley received this grant from BP along with the University of Illinois and Lawrence Berkeley National Laboratory to investigate biofuels, and this is called the Energy Biosciences Institute. And if you want to read about a lot of controversies, go on to the Berkeley web pages. And the Joint Bioenergy Institute, which we are now finalists for through the Department of Energy. So I think what I've shown you now, hopefully, is that through engineering chemistry and synthetic biology, we can actually engineer the cell to produce many different kinds of molecules. And some of these can be very powerful drugs that have limited supply and therefore are far more expensive than they need to be. Or to produce drugs that come from limited resources that we don't want to ruin our environment and cut down those resources. And of course, later on, to even do things like produce biofuels. I'd like to thank all the graduate students in my lab who did all this work. And I'd also like to thank our funding sources. This work was originally launched by work from the National Science Foundation, a grant from the National Science Foundation. And that was incredibly useful. And I just want to put a plug in for basic research, because when we started this project and it was funded by the National Science Foundation, we had no idea that we were going to use it to produce an anti-malarial drug. We had no idea that it could be used to produce a biofuel. We were really just interested in the chemistry going on inside the cell and how we might manipulate chemistry. And the point here is that you never know where research is going to lead. But fundamental basic research is so important to everything that we need for the future, new drugs, new fuels. So I really thank those of you who've supported the National Institutes of Health and the National Science Foundation. All of us are getting a lot from that. And I thank you for your time and attention, and I'd be happy to answer any questions.
Thank you, Dr. Keasling. At this time, we'd like to take some questions from everyone. By the way, what about the incidence of malaria in the United States?
Yes. So there's hopefully this microphone's on. There's almost no malaria in the United States to speak of. There's been some in the South. There's even, many years ago there was malaria in the San Francisco Bay Area, but through, actually, most of it through spraying of mosquitoes, we've gotten rid of malaria in the U.S. I don't know if there's been a recordable incident of malaria in the last few years or last few decades. But clearly, we also have access to drugs here.
Yes, sir.
Hi, I'm Colin McCormick from the House Science and Technology Committee. This is a great story, so thanks for coming and telling it to us. What I'm particularly struck by is that you did succeed in taking basic research out of the lab and putting it into an applied context. And I think, unfortunately, that's the exception rather than the rule for a lot of great research. I wonder if you could talk more about that process. What incentivized you to do that? Did the university help you out with that process? Could you just explain a little bit more how that
Sure.
went down?
So first, I'm an engineer by training, and so taking basic research into an application is what an engineer does. So to me, it's second nature and just something you would do. You're right, it doesn't always happen. But in some sense, that's what we have pharmaceutical companies and biotech companies. And I think that the growth of small companies and them being launched out of universities is actually a very important way that we transition basic research into applications. Our university, Berkeley, has been extremely helpful in that sense. For this drug in particular, university patented everything that we did, and for good reason. We do that so that no company can block us from using the technology for malaria. But the university, even though they spent a fair amount of money patenting this, paying for the patents for this work, they gave the patents away. They said, as long as nobody in this team of people, the three, university, Amyris, or One World Health, makes a profit on the drug, we'll give away our patents, free of charge. You just can't make any money off of poor people. Because the goal of the university and of patenting in the university is to get the university's technology out to as many people as possible. And so that was the mechanism that they did it. We need to see much more of this happen. I agree with you. And I think one way we do that is by encouraging graduate students and postdocs, when they leave the university, to start small companies, make it easy for them. I think in many cases, universities use patents to try to make a lot of money. And Berkeley, I think, is changing that and leading the charge in the sense of that's not the mission of the university. The mission is to actually get the technology out to as many people as possible.
Yes.
Yes, very, very interesting, and thank you. I was a former professor at the University of Michigan, so I'm very proud of the fact that you have appeared today. One of the things I would like to ask, I've just read the National Science Foundation legislation, and I'm interested in knowing whether or not there's any room in the type of structure that you have out there for young, perhaps even undergraduates, to come out and to spend a period of time enhancing their understanding. And if so, that we should perhaps provide for that within the undergraduate or the early graduate curriculum so that other individuals could see the expanse of this work because it is endless.
Right.
And I thank you today for coming.
Thanks. Yes, there are many programs. I want to mention one in particular. We have a grant from the National Science Foundation called an Engineering Research Center, and it's in the area of synthetic biology. And one of the things we do with undergraduates is we have what we call an iGEM program, International Genetically Engineered Machine Competition. And what we do is we bring students in for the summer in teams of between 5 and 10 people. And we even have high school students in the team, and this year we'll have high school science teachers in the team. And they work together as a team on a particular project. One year they engineered E. coli to take pictures, so it would actually take a picture. Another group engineered E. coli to make a banana smell. But it's not the project that's so important, but it's getting them to work as a team, right? Much of our research, we actually, especially in academics, we don't encourage teams as much as I think we should and could. And so this gets them working as in teams. And it gets them to understand the process of genetic engineering and how it's done. So I think, and then at the end of the summer, or in the early fall, they go to a school, and it's MIT in this case, and all of the teams compete. And this past year, there were 40 teams from around the world that actually competed. And because they're undergraduates, everybody wins. It's not a real competition. But they get to share their projects with the other teams. So I think it's a really innovative program. And if I would put in a plug for that if you're looking for a place to put your money and support. But I know there are many other beneficial programs, and I'm sure my colleagues from Berkeley could talk about those.
Thank you.
Sure.
Dave, one advantage of your platform is that if a drug acquires resistance, if the organism acquires a resistance to the drug, you could retool your drug potentially. I'm wondering if there is resistance to this drug and whether you've thought about ways you could potentially overcome that.
Right, that's a great question. So there's going to be resistance to artemisinin. They've shown it in the laboratory. They think there's some evidence of it in Vietnam, but they're not sure. One of the problems with abuse of these drugs is that in the past they've been sold as monotherapies, so one active ingredient. Now the World Health Organization is recommending that they be sold as combination therapies with 2 drugs. But one of the problems is that this drug is grown on little farms in Southeast Asia, and you can actually go on the internet and buy the active pharmaceutical ingredient. And anybody else can. And so there are these, what I call rogue manufacturers, that are actually selling monotherapies, and some of them even selling fakes. And in fact, the effort they go into making fakes, it's amazing. But the problem with that is that there's no control on the source for artemisinin. So you've got these manufacturers that can produce monotherapies. One of the advantages to this platform is that if you're producing it in large tanks, and it's the cheapest artemisinin out there on the market, if you can produce it cheaper in tanks than any other way, then there'll be no motivation for those rogue manufacturers to produce monotherapies, because the cheapest artemisinin will be available in tanks and only to those manufacturers who are producing combination therapies. So we can control it in that case. So this is an advantage. Back to your point about, can we use the platform to change the molecule, decorate it? Yes, that's one of the real advantages. In fact, I mentioned that after the organism produces the precursor, you can do some decorations to make all these different derivatives. And that's actually really important, because you can change, that gives you something very easy to manipulate and change the decoration on the molecule and make it so that it is more effective, not that the organism isn't resistant to it. And I should mention that there was another drug that was in phase 3 clinical trials called, I think it was OZ299. It looked a lot like artemisinin, but it was produced chemically. That just fell out of clinical trials last month. And this is a serious problem that puts even more pressure on us and on artemisinin. We have to get it out there, and we have to get it out there soon, because it's our last line of defense There are other drugs in the pipeline, but just like this OZ299, they could also fall out of clinical trials at any time. And this is a proven drug.
Just a moment.
Hi, thank you for your presentation. I'm understanding the fact that they are beginning spraying again in some of the countries in Africa, spraying with DDT. What are these, what do you know if there are any synergistic effects of the DDT with the drug?
Well, you know, I think the answer to malaria is not going to be one drug. It's going to be a combination. It's going to be spraying with DDT in limited amounts. They're finding now that it actually drives mosquitoes crazy. So they can actually spray really dilute concentrations of it inside houses. And just the smell of it. So at a point where it won't really hurt humans, but just the smell of it in the house keeps the mosquitoes away. And then bed nets that are impregnated with things like DDT or with other pesticides, and effective drugs, and hopefully, eventually, a vaccine. But the vaccine is many, many years away. And because, in part, this Plasmodium is so hard to culture, and that makes making a vaccine extremely difficult. But I think your point's exactly on target, and that is that it's not just going to be a drug. It's going to be all of these things together that are really going to get rid of malaria in the long run. And we have to be working on all of them.
Yes, please.
Will you have to go through clinical trials with the synthesized?
It's a great question. So artemisinin, as I said earlier, doesn't have FDA approval, but it does have approval from European agencies and South African equivalent of the FDA, and it's in use. But what we do have to show is the contaminant profile, because that's going to be different from what comes from the plant. And we have to show that those contaminants are extremely minute, or we have to get the purification to that point where they're extremely minute, and that none of them is toxic. Now, the advantage we have is this goes through another stage of chemistry after it's produced microbially. And so, in a sense, this is a chemical precursor, just like if you got it from oil or you got it from anywhere. It doesn't matter so much that it's biologically derived. And that's actually a real advantage, because as if you know anything about the FDA and approval, biologics are extremely difficult to get through the FDA because it's so complicated. But this can actually be treated as a chemical.
Yes, sir. Just a moment for the mic.
Thank you for your fascinating presentation. You stressed that this research has been able to bring through publicly funded. I'm curious to hear to what extent you explored other means of funding, such as college or university funding, and especially to hear the extent to which this would have only been possible with public funds because it was general research or something along those lines.
Right. So I should say that so the start of the project was through public funding, and this allowed us to get it to a stage that then we could credibly go to, in this case, the Gates Foundation, and say, look, we've got a lead on a process that will produce a known drug. Here's what we need in order to finish that off. And here's what it would mean to the world if we could do that. That wouldn't have been possible if we didn't have that preliminary stage. The Gates Foundation wouldn't have wanted to fund that preliminary stage because there's nothing, it would have been so far from an application. In fact, when we proposed it, it didn't have an application in mind, that they would not have been interested. So it's perfect for public funding, where you're investigating science. We had in mind the idea that we could produce perfumes, maybe an E. coli, ode to E. coli. But we really went to the National Science Foundation for basic research funding. And I think that's why that's so important to have that basic support from the federal government that is so far from an application that a pharmaceutical company or an agency or nonprofit would not ever want to fund it because they wouldn't ever be able to see the product that would come out of that necessarily.
Well, thank you very much, Doctor. We really appreciate this splendid lecture. In the light of the doctor's briefing, I want to remind you again, in your offices, think about the National Institutes of Health and the National Science Foundation in the next few weeks when the appropriation bills come up. We want to thank the Joint Steering Committee for Public Policy for sponsoring this luncheon. We also want to point out that our next briefing, and it's going to be a fascinating one, on June the 6th, will be on the subject of budding scientists, students from the Intel Science Talent Research. We're going to bring in several students that took place in the Intel research contest, and they're going to define and explain the marvelous projects they went through. Thank you very much.